Okay, so the ones that survive, they get to leave the bone marrow, they get to go out and explore the body and look for pathogens, viruses, cancers, other things. And when they find something that they bind to, an interesting thing happens. They start to mutate. And then the ones that bind well get selected, and they get to have more children, if you will. They get to proliferate and make many copies of themselves, but each of those copies has a high mutation rate. So you end up with a natural selection at really high speed occurring as soon as a B cell encounters a foreign antigen. And the result is that the affinity to the antigen is increased with this selection process. This is called maturation. So the B cells mature so that they are very specific and high affinity to a given antigen at a given epitope. And some of these cells exit from the process and they turn into long-lived memory cells. These memory cells can stay with you your whole life, so they are virtually immortal. We don't know exactly which cells are selected to be memory cells out of this process, and it's a hot topic of research, so I certainly am not going to try to tell you I know which ones they are. But it appears that it's not necessarily the most high affinity ones. It's not something simple like that. A diverse sampling of the cells involved in maturation go on to become memory cells. I will just say also that an important function of the memory cells is that they will respond much more quickly when they see an antigen. And so that is the purpose of a vaccine. A vaccine, the main goal of vaccine is to produce memory B cells so that when you see the virus, you don't have to wait a week or 10 days to mount an immune response against the virus. You will very immediately be able to start producing antibodies against the virus. Okay. Now this is a little bit of a jump. I just wanted to point out that B cell sequencing is a technology where you can get the heavy and the light chain sequence paired together. This wasn't always the easy thing to do. It used to be very hard to get the complete amino acid sequence of an antibody. And in particular, this company, 10x Genomics, has made an off-the-shelf technology that everyone can buy. And I guess they went public in late 2019, it looks like. And they just went on a roar, and their product is becoming very, very popular. And that is because it allows you to take a sample of blood and filter, sort out the B cells, sequence those B cells, and get all the antibodies associated with that sample of blood. Okay, so it's important to keep in mind that this technology was started becoming available for T cells maybe 2018 and maybe 2017, but only 2019 for B cells. This is really new stuff, and it changes the kinds of science that we can do, especially as bioinformaticians. And I apologize for this slide. I saw on the train ride over here that something I did messed up the colors. But what you're looking at here is a plot of the growth of the B cells. Yeah, it might even be the growth of the T cells. I'm sorry, the B cell receptor. I don't talk about B cell receptors. The T cell receptors were a bit earlier than B cell receptor sequencing. And basically what you can see, it's almost a step function. So basically there was a before single cell sequencing and after single cell sequencing, and it's around 2016. So there was two eras, you can say, of this type of work. One era where there was too little data to do anything. And then overnight, there's too much data. So now we're in the too much data side of this plot. And the B cell would be just about a year later. So basically last year. And I don't talk so much about COVID-19, a bit about COVID-19, but it turns out that, yeah, the COVID-19 pandemic and this technology, they sort of happened at almost the same time. And so there is an enormous amount of data on COVID-19 antibody sequences now. Okay. So I'm going to talk, show a few slides about something different. So I talked about the modern day now, how we can discover B cell receptors in from single cell sequencing, but it wasn't always like this. And antibody discovery, therapeutic antibody discovery, traditionally was a very, very slow process. So I'd like to just briefly go over how things used to be, say, 10 years ago, 5 years ago. So first you would identify the antigen that you're interested in, and you would immunize animals with that antigen, and you'd collect several of these antibodies. They're called monoclonal if they all have the same CDRs. And because they're animal antibodies, you don't want to use them as therapeutic antibodies in humans because our immune system will attack it. So what you tend to do is cut out the CDR regions and stick them onto a human antibody. And you can imagine that doesn't always work perfectly. And then you have to optimize it. And that's where a lot of time is spent, trying to optimize the specificity and the affinity. And of course, if it's a therapeutic antibody, you always have to worry about the safety. And many antibodies have failed because of their poor safety profile. Basically, they're toxic. They can kill you. Then you have to evaluate efficacy. And so this process is long. And I want to just show you two examples. So two of the real stars in antibody discovery are Honjo Sensei and Kishimoto Sensei from Kyoto University and Osaka University, respectively. And they both have made major discoveries in therapeutic antibody research. Honjo Sensei for checkpoint inhibitors, antibodies that bind to cell surface receptors that will allow us to have a new approach to cancer therapy. And then Kishimoto Sensei, who discovered the interleukin-6 receptor and then developed an interleukin-6 receptor blocking antibody. And so for PD-1, the discovery was in 1992, but it wasn't until 2014 that the actual antibody binding to PD-1 was on the market. That's a long time. And then it was years later that it was actually making a lot of money. So if you think you're going to get rich by making an antibody, you have to be healthy, like Honjo Sensei, and live a long time because it takes a long time to make therapeutic antibody. And IL-6 was even, I think, more extreme. So 1988 was the interleukin-6 receptor discovery. And really, it's only kind of recently that the sales of this antibody have been significant. And so this is one of the reasons that pharmaceutical companies don't just like to pursue antibody therapies without thinking about it a lot, is that it's slow and expensive. So not everyone is going to make a blockbuster drug. But what I'd like to argue, what I'd like to argue is that some of the technology that I'm going to describe today could accelerate this process. Okay, so this is just a peek at some of the data. I told you that COVID-19 has resulted in an enormous growth in the antibody sequence data. And you can see that by this blue bar here. So that's this year. This is the number of sequences. And I list several other viruses that have been studied from the point of view of antibodies in the last few years: HIV, influenza, I already showed you influenza, hepatitis B virus. And if you add up these bars, basically you get to 20 million sequences in 2020. So right now in our lab, we are playing around with this 20 million sequences just for 4 viruses. Okay, so you can imagine the amount of data if you look at not only these 4 viruses, but other pathogens, not only viruses, and then other diseases, not just infectious diseases. So it's an enormous, unexplored world of immunology and disease at the interface, and that interface is these antibody-antigen interactions. Okay, so that's the end of part 2. Now, part 2 was a kind of wandering, but I'll try to summarize. So collectively, antibodies have a kind of immune intelligence. They have some of the properties that we normally associate with neurons. They can distinguish self and non-self, which is non-trivial. And that would not be possible if you didn't have this inefficient method of generating random antibody, B-cell receptors, and then removing the ones that happen to bind, so this training that happen to bind to self-proteins, and then keeping the ones that don't, and then training them to bind to foreign antigens. They learn to bind with high affinity and specificity through this maturation process. And then finally, they remember the things that they have bound to by this process of memory B cells. So altogether, it's a pretty sophisticated system. Okay, this high throughput antibody sequencing at the single cell level really marks the beginning of a new era, and we can do some new types of science. And there's definitely room for all kinds of bioinformatic methods, both structure and not structure based methods in this field. And hopefully, some of these new methods are going to allow us to do therapeutic antibody discovery in a more efficient and sophisticated way than we did in the past generations. Okay. So that gets us to part 3. Okay. So part 3, I'm going to talk about high throughput antibody modeling. I already told you about low throughput antibody modeling in this contest that we entered. And I hinted that fast methods could be just as accurate as slow methods. So our latest software is called Repertoire Builder, and it is a new approach different from the COTI antibody builder that I introduced before. The main feature of Repertoire Builder is its speed. It is possible to input 10,000 antibody sequences and get out the results in less than 30 minutes. And the reason that it can be so fast, I don't have time and I don't think you're interested in all the details, but the key difference between what we did in COTI antibody builder and Repertoire Builder is that we do all of the decision making for the modeling in terms of sequences, not in terms of structures. So we don't use like an energy function that scores the structures. Rather, we use feature vectors. Those feature vectors are based on multiple sequence alignments. So this allows us to not have to touch the structure until the very end at this last step here. This mouse is behaving strangely. I don't know why. Well, anyway, the last step, we take the pieces from our templates and assemble them. In the COTI antibody builder, we tried to score each structure very quickly. So COTI antibody builder was fast, but repertoire builder is hundreds of times faster. And so that allows us to do massive or high throughput antibody modeling. And the great thing about repertoire builder is that, at least to date, in our hands, there is no apparent sacrifice in the accuracy. So this plot right here, for example, these are B-cell receptor, CDR, heavy chain, CDR number 3. So this is the one we usually care about the most. This is a program called Lyra. These are the errors here. This is a program Pigs Pro, a body builder, and this is repertoire builder here. So you might say, well, the shapes of these distributions, these violin plots, look quite similar, but certainly repertoire builder is no worse than the others. And you might wonder, well, what's this one here? This is the ideal. This would be the case where you're allowed to pick any template from the PDB and try to get the one with the best RMSD, so that if you could see the accuracy of your structure. The reason I include it is to emphasize the point that if you're doing template-based modeling, the perfect answer isn't 0 error. The perfect answer is some error. And so we're not as good as ideal, but we're better than the other programs, or no worse than the other programs, and quite fast. And it also works for T-cell receptors, etc. So we're kind of happy with that. And it seems that this year, some other groups around the world are using repertoire builder for their COVID-19 work. So that's nice to see. Okay. So let's summarize what I told you about antibody modeling. So antibody modeling can be very fast. And there are other fast methods, not just ours. So Lyra, which I showed you on the previous slide, is actually a bit faster than repertoire builder. So we're not the only ones that are fast. A-body builder is actually quite fast. It's not as fast as repertoire builder, but it's close. Repertoire builder appears to be both fast and accurate, which is what we want. And so this comes back to the lesson that I learned in the antibody modeling contest that I want to repeat, which is sometimes you don't need to use the most complicated algorithm. Sometimes you don't need, we're in the K computer building here. Sometimes you don't need a K computer to solve your problem. It's nice if you can always use the K computer, but not everyone can. And sometimes you can do a lot with fast methods. And this is one of those cases. That doesn't mean that molecular dynamics is not appropriate for refining these models. We're actually doing that right now. So I think we have to remember to use the computationally intensive tools where they are most best suited. And this is an example where probably template-based modeling can get you very close to the answer, but it can't do anything after that. So then we have to use a more physics-based approach to optimize the structures. So finally, the last point is, so we can now make lots of antibody models. Who cares? Actually, that's not in itself a good thing. Just making models is not a very important goal. Maybe for bioinformaticians, but not for immunologists or people trying to develop vaccines. We need to figure out how to use these models to understand antibody function. And in the context of most of the research being conducted on antibodies, that means predicting their specificity, predicting the epitope on the antigen that they target. So this is the holy grail. And I'm not going to tell you that we can do it today, but I'm going to try to indicate a kind of direction that we can take to get closer to that goal. And that direction is, I believe, antibody clustering. So let me just take a drink. Okay. Antibody clustering Doesn't sound very exciting. And frankly speaking, it's not very exciting, but I'm going to try to make it slightly interesting at least. At the core of any clustering algorithm is a distance function or a similarity function. And it's no different for antibody clustering. So I'll start with this slide with how our software called Interclone approaches the problem. So just like with Repertoire Builder, we will treat the antibody as a structure that we can use to make a feature vector. And we're going to use that feature vector to train a classifier. And the goal of the classifier is to say whether two antibodies are targeting the same epitope, okay? So in general, if you pick two antibodies at random, they don't target the same epitope. So you could actually get a pretty good classifier if you just said false every time. You'd have a pretty high success rate because most antibodies don't target the same epitope. So obviously we cannot just use that approach. We have to reward the times that it's right and try to minimize the times that it's wrong. But the basic core of Interclone is just here. We have these repertoire builder structures of antibodies or T-cell receptors, and we try to derive some features from them that will be somehow correlated in a way that we find the antibodies that do target the same epitope, but that are not trivially the same sequence of amino acids. The approach we use is really similar to what you have in your cell phone, your keitai. Most of us have photographs on our phone, and most of our phones have software that organize our photographs for us. Some of them are very sophisticated, but even the most basic AI software can tell a face out of a photo, can pick out a face, and moreover, can group those faces together and say, this is mom, this is you, this is your family. So the way that works, actually I'm not an expert, so if you're an expert, I'm sorry if my explanation is bad, but there are certain landmarks on the face, and then we can connect those landmarks by lines, and the vectors connecting those landmarks creates a feature vector. So it's not so different from antibody. This is the paratope of an antibody. There's different CDRs on the antibody. Those CDRs are located in different locations. They have other properties, the chemical properties, and also their lengths and shapes. And we can connect those and make a feature vector. So I'd like to illustrate how this works with a real classic antigen system, which is HIV. So HIV is probably either HIV or influenza. One of those is probably the most studied antigen source of all time. So we have many, many anti-HIV antibodies in the protein data bank, and they can be classified according to their epitope here. The CD4 binding site, V2, V1 site, V3 site, fusion peptide, GP4, EMPER, etc. And so there's a database called CatNap, which collects these antibodies and groups them according to these epitopes. So what we wanted to do was train our clustering software called Interclone on data that is not on this slide, other antibodies like influenza, etc., and then see if Interclone could put the CatNap antibodies in the right groups. And it turns out that Interclone did quite well. So what we have here on the left side are the clusters of antibodies. So each circle is an antibody. And this is, oh, and then the colors here are shown over here. So these are the different epitope groups. And you can see that mostly the ones with the same color are put together in clusters. So overall, it's definitely doing better than random. The left side is if we just use one of the chains of the antibody, and the right side is if we use both chains. And so you could see that the results indeed are different. In particular, you see this big group here that we could only put together if we have the light chain. This was all spread out over here when we had only the heavy chain. And so it's kind of interesting. And of course, it seems to make a mistake also here. That doesn't look right. It's not perfect. That looks like a mistake there. There's a mistake there. But overall, most of the big clusters look to be correct.